Okay, for those that don't know, I'm Janell Richardson. I am in research and development at Taconic Biosciences. We're a preclinical model company that specializes in everything from the tried and true, like B6, all the way up to our humanized immune system models. You know, we thought there could be a similarity because if many of you remember the fall meeting, Frank gave a great presentation on Fc gamma receptors, both their importance within especially biologic drug discovery, implications of binding, low affinity versus high affinity, activation versus inhibitory, Fc engineering, honestly going into a realm that to be honest with you, I certainly do not have the proficiency or expertise that someone like that or the possible working group centered on Fc gamma receptors has. What I ask of you and this audience today is really more of an engagement. It's not going to be me presenting on all of the nitty-gritty behind these models. It's more, these are options, these are models that we have. Could you help us with understanding relevancy or not relevant to the market, where assets could be developed, interesting questions we can ask, and really be able to have an opportunity to determine, could this model help, right? Could it help to provide a more translatable outcomes? So I really encourage this to, again, be a dialogue. As Elise already prefaced, I really have only 2 slides. You are more than welcome to go to the original manuscripts. Each of them have a hyperlink in the slide deck that was attached to this meeting. So each of you should be able to easily download these slides. You can just click on the word manuscript and it should send you directly to the original journal article. These models were created with our partner, CIA, which is based out of Japan. In particular, the investigator there is Takeshi Takahashi, who is the last author on both of these papers. And basically, as the first bullet point already hints at, it's a super immunodeficient model, the NOG, where there are 2 mirroring genes that have been knocked out. And FcyR1G, which is just known as kind of the backbone of the Fc gamma receptors. It is an activator, right? Fc gamma receptor 1, and the FcGR2B gene. So there are 2 genes that are knocked out. This does differ from a paper published in 2015 with an Fc gamma receptor KONSG that basically used a model by Rafferty that only had FcyR1G knocked out. So just want to make a distinction between those 2 potential models. So this one has both genes knocked out because both of these have large implications in terms of the mirroring innate effector cells participating in the Fc basically binding of human biologics injected into these animals. So the whole goal was for CIA to say, hey, we know about one of the problems, especially it becomes relevant to the next slide. But for this slide, the parent model, which is taking these 2 genes, FcyR1G and FcGR2B, and providing a model to which the mirroring innate cells can no longer participate in things like an ADCC-based efficacy response, right? So trying to, again, from a chimeric model, which we know that these often are, right? Humanized immune system mice are always a part mouse, part human. You can't get all human, mouse won't live. You can't get all mouse, otherwise why would you bother? So in developing and increasing the, again, translatability, it's how do we try to prevent the mouse part of this model that is still required for the model to live, but not to confound results that we have traditionally seen. And so an example of the parent model, which is the Fc gamma receptor NOG, is engraftment looking at 4 different CDXs, and you can see the CDXs on the right-hand side. They basically looked at a model where they used Opdivo and asked the question, is there a difference in checkpointer response between an Fc gamma receptor KO NOG versus a parent NOG? Now, one thing I will mention is that, unfortunately, Fc gamma receptor KO itself does not contribute to altering the half-life in these models, right? So if you remember from Frank's beautiful presentation in the fall, there's another transporter involved that is critical in the half-life of monoclonal antibodies in particular, and that's the Fc neonatal receptor. So I will just preface this by saying that the PK, or the half-life expectations of the model, should be viewed as similar to the original parents, which tend to be accelerated. So they looked at and compared between an Fc gamma knockout and basically the original NOG. Asked the question, is there any differences in overall engraftment? They found a small increase in the degree of chimerism, but I would say it wasn't huge. They really did not see a drastic change in the overall kinetics of the engraftment of the model. Again, they put in these CDX tumor lines and asked the question, in the presence of Opdivo, what kind of response rates do we get? And you can see between the blue, which is the saline vehicle, it should be more like a isotype, but a control arm nonetheless, versus the Opdivo treated arm. You can see that clearly one of the CDXs is a non-responder regardless. So these are all PD-L1 positive based cell lines. But there is a discrepancy between basically the top graphs that you see, which is in the Fc gamma receptor KO NOG, same donors, and the bottom graphs, which is the HUNOG. Clearly, there is at least a separation between the red and blue lines in the top graphs compared to in the bottom graphs. And that's kind of reciprocated in the overall results. This isn't a define or really saying with this model, checkpoint inhibitors become clean and become much more responsive. Again, I think it's just preliminary work that I would just encourage us to use as a bounce off for how do we improve upon, how do we answer questions that you guys have, and possibly, again, also collaborate, right? We have this model, it basically, I would say is near to our production levels within the next few months. And we really want to better understand where its niche is and whether there could be implications beyond just, for example, these two models. So all questions that we have. Next slide, Elise. So this is the model that I would say we have a little bit more understanding and grounding in terms of its potential relevancy. So this is the Fc gamma receptor KO NOG that in turn was crossbred to the IL-15, the human interleukin 15 expressing NOG, in turn creating an Fc gamma receptor KO that expresses IL-15. This becomes especially relevant to NK cell mediated ADCC activity. And what do we mean by that? What I mean by that is a classic case where you see a HER2-based, drug in a gastric CDX with basically a HEMI, so HEMI for the Fc gamma receptor KO, and then a homozygous knockout on the right-hand side. And what you notice in particular is the difference between the black and the red. So these models have absolutely no human cells in them. So we have a tumor on board and we have a drug targeting that tumor, but we lack the human cell that in turn should facilitate efficacy. Yet in a HEMI model, we're showing just that, right? We're actually showing that the tumor is decreasing in size with the presence of this antibody on board. But yet the cell required for this response is not there. And that's because these are mirroring innate effector cells that are binding to this antibody that are actually causing this type of response. It is not a human-based led response. And you compare that to the right-hand figure, which is the same exact experimental design, but with a homozygous knockout. They do not have the Fc gamma receptor 1 or the Fc gamma 2b receptor. So certainly the premise here is things like NK cell mediated efficacy and being able to eliminate that confounder in terms of a false positive. There's also the potential to deal with false negatives as well in either of these models. So I kind of want to stop here. So again, the very last bullet point for those that would like to go to the original journal article, because certainly there's a prestigious amount of material that you can read through, is the hyperlink on the manuscript. But with that, I kind of want to move to, again, a dialogue with this group that clearly has sometimes quite a bit more experience than we do with Fc gamma receptors. The implications to your, to the work that you do, again, questions that we can ask and provide data to that could help in turn say whether it has particular use or not. So with that, we also have some other questions that certainly, if you have the time and the ability, we would love to just know a general, your basically opinion and answers to those.